Hello everyone. So we are going to look into glycolysis. So glucose has three major fates. It can either enter into glycolysis and give pyruvate, ATP and NADH, or it can enter into pentose phosphate pathway and give ribose 5-phosphate and NADPH, or it can be stored as starch or glycogen. In case of plants, it will be stored as starch, and in case of animals, it will be stored as glycogen. So glycolysis is a linear pathway and it consists of 10 steps. First 5 steps are preparatory phase and the next 5 steps are payoff phase. So in the preparatory phase, first the glucose has to be charged for it to enter into glycolysis. So this is done by the enzyme hexokinase, which transfers a terminal phosphoryl group from ATP to glucose at the 6th position to make it glucose 6-phosphate. For this reaction, magnesium ion is an important cofactor, as hexokinase binds to magnesium ATP and not ATP alone. So this glucose 6-phosphate, which is formed at the end of the first step, is now isomerized into fructose 6-phosphate by the enzyme phosphohexose isomerase. This is a reversible reaction. Okay, so then the fructose 6-phosphate is converted into fructose 1,6-bisphosphate by attaching one more phosphate at the first position of fructose by the enzyme phosphofructokinase 1. Even here, the phosphate group is given by ATP. Then next, fructose 1,6-bisphosphate is cleaved into 2 triose phosphates by the enzyme aldolase. The first triose phosphate that is formed is glyceraldehyde 3-phosphate and the second triose phosphate is dihydroxyacetone phosphate. Here there is one point. Till fructose 6-phosphate is formed, it can enter into any other cycle apart from glycolysis. But once it is converted into fructose 1,6-bisphosphate, this molecule has to enter glycolysis and no other cycle. So this step here, which is catalyzed by phosphofructokinase 1, is a committed step. Once fructose 1,6-bisphosphate is formed, this molecule has to enter glycolysis. Okay. So when it is cleaved into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, only the molecule glyceraldehyde 3-phosphate can enter into the next steps of glycolysis. Dihydroxyacetone phosphate doesn't enter into glycolysis. So the enzyme triose phosphate isomerase converts even the dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. So there will be 2 molecules of glyceraldehyde 3-phosphate going into glycolysis. So the next phase will be payoff phase, and this will be the 6th step of glycolysis, where 2 molecules of glyceraldehyde 3-phosphate are converted into 1,3-bisphosphoglycerate by the enzyme glyceraldehyde 3-phosphate dehydrogenase. In this step, one NADH molecule is produced per molecule of glyceraldehyde 3-phosphate. So here, because there are 2 molecules, 2 NADH are produced, and here the phosphate donor is an inorganic phosphate and not ATP. So 1,3-bisphosphoglycerate, which is formed, is then converted into 3-phosphoglycerate by phosphoglycerate kinase, and in this step ATP is formed. So one phosphate from 1,3-bisphosphoglycerate is transferred to ADP to make it ATP. Because the phosphate is coming from a substrate, it is called substrate level phosphorylation. 3-phosphoglycerate is then converted to 2-phosphoglycerate by phosphoglycerate mutase. The enzyme enolase removes a water molecule from 2-phosphoglycerate to make it phosphoenol pyruvate. Then the enzyme pyruvate kinase converts phosphoenol pyruvate to pyruvate. here one more ATP is formed. Even this is substrate level phosphorylation. As the phosphate group is coming from the substrate to ADP to form ATP. So by the end of glycolysis, you'll have 2 molecules of pyruvate, then 4 ATP and 2 NADH. So as you can see here, there are 3 chemical transformations that are taking place. One is degradation of glucose to yield pyruvate, one is phosphorylation of ADP to ATP, and the one is for formation of NADH. So the net equation will be glucose plus 2 NAD plus 2 ADP plus 2 PI gives 2 pyruvate plus 2 NADH and plus 2 ATP. Okay, here pyruvate, NADH and ATP molecules are important.